Thank you. It's a great pleasure to be here today, and ironic in some ways that I'm talking about the amazing research going on in Boston, and yet I'm about to depart for the West Coast. Although my intention is to create a Bay Area Boston bridged Autism Consortium so we can continue the collaborations that have been going on here. So today's talk is a little bit different from what I'm used to doing. I was essentially approached by Dee Dee and Stephanie with the idea to try to develop a connectome and to understand in a network sense and through bibliometric analysis the rise of research over the time frame around the formation of the Autism Consortium through to the present day, and to be able to understand what that means, but also to develop a resource that we can utilize as a community to look back over the history of research in autism in Boston and to look ahead towards new potential connections that can be particularly innovative and that can compel and accelerate new research in the area of diagnostic detection and prognostic marker development and other things in autism that are badly needed for the field. So I'll start first by saying that this is entirely a joint effort between myself and my student, Marlena Duda, who's somewhere in the audience. And so everything I'm putting forth here is entirely things that we've worked on together over the last several months. I guess I'll start with what everybody already knows. Autism is rising rapidly for lots of potential reasons. 1 in 50 is a recent report that came from the CDC parent calls. And so this is a self-reported incidence rate. The more typically reported incidence rate is 1 in 88, listed on the slide here. But every year, 1 million children have symptoms that raise concern of risk for autism. And early diagnosis, we know, can do a lot for the benefit, have a huge amount of benefit and significant gain in terms of developmental abilities in the child. And yet, we're still intervening too late. Let me see if I can figure out how to use this thing. Okay. Cost is significant, and this is a significant toll on the healthcare system. And so we as a community in Boston in particular, no doubt have been working together to try to solve these particular problems. This is a public health epidemic in many ways. It's a crisis, and I think solutions are badly needed so that families can receive the care that they particularly need. So we know it's a high incidence disorder. It's defined by impairments in 3 domains. A language, restricted range of interests, and social interaction domain. There's a significant male bias. I'm sorry, I'm a little behind. My slides are advancing faster than the ones on the screen. And it's diagnosed today through behavior almost exclusively. The reasons for some of our lack of traction to date, although we've had a significant amount of gains in autism, has to do no doubt with the vast complexity, not only in the behavioral landscape of autism, but also in the genetic landscape of autism. Anywhere between 200 and 1,000 genes may be implicated, and its behaviors, the behaviors of autism, as we define it today, overlap significantly and are comorbid in often cases with many other conditions, neurological and otherwise. So what the Boston Autism Consortium has done and many other consortia have done is try to create an integrative center type approach, a virtual center, if you will, that enables us to begin to create a pipeline through detection in a clinical facility to data generation. And this might be many modalities of data generation, DNA, RNA, protein, behavioral characteristics, and others, to develop ways to interpret that and to have clinicians interpret that in efficient timescales that fit within clinical time windows to enable treatment to enable the planning for a particular child's treatment and to track that child as the child progresses through treatment. So when approached by the Autism Consortium, they essentially were looking for some way of creating a connective network that's not unlike many social networks that exist today. Facebook, of course, Twitter, LinkedIn, YouTube, Flickr, all of these things we use, I think, every day, at least I do, to connect with friends and family and to learn about new people, find lost colleagues, and make new connections that enable either more fun or better research or many other things. And so what we have done, Marlena and I, again with the help from Dee Dee and Stephanie, is to create the infrastructure necessary to provide a complete Facebook-like, LinkedIn-like connective network for the Boston area researchers focused in autism. So today, I plan to show you that in screenshots and promise that it will soon, at some point, be live on a website that enables you to peruse and search it and to find connections and to study them yourselves. So let me first start by explaining how we have begun to develop this network. So this has entirely been a bibliometric mining experiment in which we've created natural language processing approaches and procedures to mine the entirety of published literature for specific elements that are relevant to the Boston-based autism research and autism. These natural language processing algorithms enable us first to filter the entirety of Medline and PubMed down to the compendium of publications on autism and in the world, and that's currently, when we last did this, 42,153 publications that have been published worldwide and have been deposited in PubMed. We can mine that using NLP to find specific elements that are indicative of the Boston-based autism research community. Of course, that includes searching for, using fuzzy matching, for example, for Harvard Medical School and other institutions within the Boston area, to be able to develop a repository, a filtered repository that contains just Boston-based publications. And then we can go further into those through additional NLP to find specific individuals and to create from that a network that contains nodes and edges, where the nodes are represented as are individuals and the edges are the connection between those individuals. And that connection represents, in effect, a peer-reviewed collaboration, a peer-reviewed event of discovery that was jointly had by those two individuals. And when we do this, we can develop a full network and begin to study that network over time and also in its totality. So that is what we have done and what I'll present for you today in the time that I have, with some very simple questions that we can begin with. What is the shape of the connectum? How big is it? What does it look like? What are the connections? How has it changed over time? How has that shape changed over time? What's the rate of change of that shape? And what does that mean with respect to increases or decreases in relevance of interdisciplinary collaborations, and what are those interdisciplinary collaborations doing for us as researchers in the field, but in terms of understanding the molecular pathology, characterizing the molecular pathology of autism, and increasing our understanding of potentials for clinical impact, but also what is this interdisciplinary collaborative sphere of research in Boston doing for our families? Are we getting to the point where we're starting to give back? And is there going to be a lot more on the horizon and in the near term that give back to families? All right, so first I should define, this is the first network that we created. We wanted to take a look at all networks in Boston in and around the formation of the Autism Consortium in 2004. So the nodes here are blue, the people nodes are blue, the institutional nodes are green. Yellow edges indicate an edge between two individuals, and blue edges indicate an edge between an institution and an individual. So in 2000, this is a fairly sparse network, and there are lots of singletons off in the corners that are not connected to the main network. At this particular point in time, based on our bibliometric mining of the entirety of Medline and PubMed, there are 57 contributors and 62 collaborations. So, relatively modest. Of course, this is early days for autism research. In 2002, this has increased significantly. 95 contributors have come on the scene. More institutions have come on board that weren't there previously, but more importantly, there's a deeper interconnection network here that indicates a higher level of collaboration is occurring already at 2002. At 2004, we have a huge increase in both connectivity and contributors. This is at the time of the formation of the Boston Autism Consortium, and this particular network contains 131 contributors, 400 collaborations, and an incredibly and statistically significantly more connected network than 2002. So we've jumped ahead and we've advanced significantly by this time frame in terms of connections. In 2006, this is not surprising probably, and what I'm about to show you is there's been an incredible rise in activity over the time frame since the formation of the Autism Consortium here in Boston. This is no doubt in part because of the Autism Consortium's efforts in terms of networking researchers, but it's also a testament to the interests of many in the field and to the formation of other interest groups that have raised awareness, that have brought the attention of this important heterogeneous disorder and the need for intelligent scientific investigation forward, and have enabled others to begin to contribute. So 2006, we now have 161 contributors, 306 collaborations, and again, a lot more dense connectivity here, statistically increasing in significance relative to 2002 in particular. Now in 2008, there's a huge change in connectivity, where we have now 249 contributors from the previous 161. We have a big jump. The biggest note to make here on this particular slide is that we went from a relatively small number of collaborations, 306 interdisciplinary, well, I won't say interdisciplinary, just say collaborations at this point, to 1,188 collaborations in 2008. That's a huge jump, and this indicates that the field is becoming, at this time in 2008, much more collaborative than it had been previously. 2010, the trend continues. Continual increase in activity. By the way, the sizes of the nodes are indicative of the number of outgoing edges from that node. So bigger nodes mean more people, essentially. So there's more researchers that are coming on board and starting to participate, probably through, and we can prove this through the analysis of the Connectom itself, through collaborative interests. So individuals who were working in a field that was previously tangential to the interests of the autism community got brought in by somebody and asked to participate. And that's what's happening more and more. 325 contributors, 1,579 collaborations. 2012 is maybe hitting the asymptote at this point of collaborations. We're at 1,485 and 297. But nevertheless, it's a sustained level of interactivity and collaboration that now exists across Boston Autism Consortium members and Boston Autism researchers. I should caveat this by saying that in the beginning, we began with a list of individuals provided by Dee Dee and Stephanie who are participants formally named on the Autism Consortium's website, or part of the Autism Consortium formally. We went with that and also went beyond that using NLP to find other Boston area Boston researchers to be able to enable individuals in this room the opportunity to identify those people out there that they don't already know, who aren't already part of the Autism Consortium, and also provide the Autism Consortium with a mechanism, a software mechanism essentially, to identify individuals who should be in the network that aren't currently in the network. So this is an opportunity to grow the Autism Consortium's researchers contributor list, and to be able to create more energy in the area of autism research that will have exciting, no doubt, outcomes and opportunities to be able to advance the field. So I just wanted to give you we're not done yet, of course. We haven't been able to tally up all the 2013 publications, but at this point, there's a huge number of contributors, a sustained growth of collaborative research. There's 1,780 collaborations in this particular year. So there's a huge increase. Excuse me. So let's look at this from a little bit more of a graphical perspective and mathematical perspective, and also to look at this within the context of what's going on elsewhere. It's nice to know what's going on in Boston, and there's no doubt that's a lot. But what does a lot mean? What does it compare to? What do we use as an outlier for comparative analysis here to understand, is there a difference, or is this the baseline? It turns out that Boston, in general, has been outpacing, in terms of activity, numbers of publications, New York, San Francisco, and Philadelphia. Other areas where there's arguably significant levels of activity in autism research. And with me going to California, it should increase in the San Francisco Bay Area. and citations are also outpacing in terms of the impact. So this is actually meant to indicate impact. The impact of the publications coming out of this research sphere that I just described, the Connectom, and the Boston activity and energy, is significantly more impactful in terms of numbers of citations than the numbers of citations that are being made on publications coming from Philadelphia, New York, and San Francisco. So this is good news. It's not meant to say we're better, but it's just meant to give people a perspective that this is actually meaningful. This Connectom isn't just a ball of spaghetti. It's actually working in many ways, and it's providing, arguably, it's providing the medium for more impactful research, more citation-worthy research. So along the way, I just wanted to highlight some of these things that you all probably know about. What happened in the time frame as this Connectom rose and as the number of connections increased. We have GWAS studies, of course, that are coming out of Boston, the 16P study. We have studies on from 2010 providing a first look at use of genetic information for diagnostic testing and CNVs. We have electrophysiological research that's emerging that's indicating the potential for the use of EEGs as a biomarker for distinguishing between high-risk and low-risk individuals. And more recently, we're starting to learn from this particular community, about 2-hit loss-of-function mutations and their potential impact on the molecular pathology, of course, of autism, but potentially in their use as diagnostics for a subset of the population of individuals affected with autism. And I'll talk a little bit more about the impact of some of the research that's come out of the Connectom in a few slides. So the annual rate of collaboration is another thing to take a look at. We have co-authorships have been increasing, but also collaborations here defined as a joint publication between two individuals across the full set of publications that year. And so in many cases, there's many more collaborations than there are just pairwise interactions between individuals in the Boston area. And so this is just to really stress again that it's not just that there's more authors and more impactful research. There's an increasing density of connections that is happening in Boston. So let's take a look at the total of the last 12 plus years of research. There's been 1,117 papers that we've been able to find and to annotate using our natural language processing approaches. This amounts to 20,000 pages of results, roughly, and that's a significant corpus of data. 3,825 total authors, 438 authors with greater than 3 publications, particularly prolific authors. There's a lot of those in this community. 6,184 co-author pairs and nearly 10,000 collaborations. This is huge. And that's the sum total across the Connectom from 2000 to 2013. Now, it was interesting, I was trying to understand what 20,000 pages might look like in a physical sense. And I currently am in a library, so I thought I would stack up encyclopedias until Marlena said, why don't you just Google it? And so I did, and this is entirely just Google accident, that the first hit that came up was this. I guessed 7 feet, by the way. I thought it might be 7 feet. It turns out it is 7 feet. But this is actually a picture from a tweet from Senator McConnell complaining about Obamacare. This is not meant to be political, but apparently this is Obamacare, according to Senator McConnell. Anyways, but it was so ironically that came up as the top hit. You can do your own Google search, and you can trust me that it's actually accurate. But that's a huge amount of data, results, and interpretation that has been generated over the last 13 years, and this is tremendous. So who's who in this in this endeavor to find out what's going on with autism. These are the top publishing authors. They're not all the top publishing authors, so if you're not up here, I apologize, but I just wanted to get some faces up on here to explain that many people in this community have been contributing in very significant ways, and these are some of the top ones that came up from our Connectom evaluation. Helen Tager-Flusberg, Chris Walsh, Charles Nelson, Mustafa, and the others you can see named there. So Helen, 55 publications. Joe Biederman, 28, and so on. These are tremendous levels of activity. They're contributing. They're also collaborating. These are our top collaborators based on the Connectom. And again, this is a website. It's meant to be something that you can search. So I'm just showing you some of the ways in which you can search it. And so we search it in this way. We come up with our top collaborators. These are the individuals who are playing nicely with others and who are working hard to collaborate with their friends. Most co-authors, so we have 118 co-authorships with Jim Gusella, 250 collaborations. This is a tremendous amount of collaborative energy. And as you can see, I mean, just try to wrap your brain around. I don't even know how they have time to have that many collaborations. It just doesn't seem fathomable to me. But nevertheless, it happened, and the Connectom told us, that it happened, and this is entirely an objective data mining experiment. This is the outcome, and I think it's a very exciting outcome. So we can look at the number of collaborations, let's say, between these individuals. I'm not sure if you can see these edges, 16, another number, 13, 18, and so forth. So just to show you that these connections are being formed, and these represent, in many ways, subnetworks of the Connectom that are particularly rich in activity. Top collaborations, we have top collaborators. These are the top collaborations, the total number of pairs of individuals who are working together with great regularity. And this is just to show you that these data are available, and you can search them and begin to use them to understand what has happened historically and what might happen in the future, how we might change things, how we might contribute to the particular collaboration that's ongoing between these two pairs of individuals that we have a lot of alignment with in terms of research interests and so forth. So all of this is exciting, but what I want to start to get into now is whether these collaborations are simply within lab type collaborations or like-minded people working together. We have the geneticists and the genomicists working together and not playing with the clinicians, not playing with the clinical psychology community and phenotypic experts and others. Is that the case? Is that what's the makeup of the Connectom, or is it more that it's interdisciplinary collaborations that have been growing, and not only in number but in diversity, different types of interdisciplinary collaborations? So to do that, Marlena and I created another set of algorithms to be able to quantify interdisciplinary collaborations through essentially keyword searching within the biblion. So we can develop an ontology, a structured vocabulary for all the keywords that are associated with all the publications by all the Boston Autism Consortium and Boston area-based researchers. We can then we can structure the vocabulary in a variety of different ways that enables us to look for patternings and to be able to characterize automatically and algorithmically an individual by their keywords. So we can say a person is this type of person with respect to their research interests as opposed to that type of person with respect to their research interests. So the best way to illustrate this is to use these things called wordles or word clouds. So we can take a word cloud from one researcher and then compare that algorithmically to the word cloud or wordle of another researcher to find differences in dominant patterns, differences in big most frequent keywords. So physiology versus disorder. When these two individuals collaborate, I don't know that they ever have, an interdisciplinary collaboration from our perspective has been formed. And so the algorithm that we wrote will look for these and then count them. And then we can use that to quantify the extent to which there's been interdisciplinary collaborations over the time frame of 2000 to 2013. So there has been a lot. There's been a huge number of interdisciplinary collaborations. Again, this is where individuals are collaborating in totally different fields, in fields that are genetic in nature versus physiological in nature, metabolic, behavior, and so forth. So this is just to show you there's been a big spike. This actually mimics the pattern and distribution of the previous slides, but it's a little bit more striking in that you have ups and downs, essentially. Not as much difference between Boston and the other areas, except at this particular time frame we have this huge increase in activity of interdisciplinary collaborations as we've defined them. This is interesting to look at, and again, it's something that we can mine the Connectom. You all can mine the Connectom as it becomes a website to do your own searches, to look for your own potential for interdisciplinary collaborations. Excuse me. So moving on. Tim is such a photogenic guy, but I couldn't find a picture for him. So I just put the Android image up in his box. This is Tim Yu's Wordle. So what happens when you take somebody like Tim and someone like Lenny and Chris and Eric and put them together and begin to understand what this ecosystem of collaboration can generate? And so over the next few slides, I want to talk about impact. I want to talk about the impact of interdisciplinary collaborations. And I'm starting with a publication that emerged in 2012 that is represented by this interdisciplinary subnetwork, which I show in a zoomed-in version here. This interdisciplinary subnetwork, represented by some of the individuals' word clouds that I just showed, resulted in an incredibly impactful paper in Neuron. This is a particular paper by you and others that you probably can't see, which is one of the most impactful exome sequencing papers for autism to date, that took an incredibly innovative approach that combined in vitro functional analysis and exome sequencing, which has, to my knowledge, not been done with much regularity at all before, to be able to identify 3 genes of significant interest in multiplex families. A gene called AMT, sign, what was the other one? PEC7 and sign 1. So anyways, AMT is the most important one, at least because I'm going to highlight it here today. This particular gene has an effect on phenotype that causes a neonatal metabolic condition that is life-threatening in many ways. Its discovery here through this in vitro functional analysis is one that is particularly informative because it provides us with not only an illustration that the use of mixed evidence, genome-wide linkage, exonic variation, biological validation, and clinical relevance to existing syndromes can convincingly implicate a rare variant. And it also highlights the potential for better genetic diagnoses that have significant potential for interventional treatment in at least a subsection of ASD. So an incredibly impactful outcome from interdisciplinary collaboration represented within the Connectom. Borrowing from a recent review in the same issue of Neuron by a review by Geschwin and others, the global this is actually an interesting thing to look at because I wanted to, we need to know where have we been, what do we know, and what don't we know. This particular study is exciting because although a lot of the variants can be explained by common variation, the effect size of any individual variant in that particular subsection of the pie is very small, whereas these loss of function, rare variants that are being discovered through clever, innovative, and integrative approaches like the one I just illustrated are likely to have significantly higher effect sizes and therefore can be used in ways that are significantly different and potentially more clinically translatable than the ways in which we can use common variants. Of course, there still is a big part of the pie that remains unexplored and unexplained, and this will be something that I look forward to seeing getting explained by interdisciplinary collaborations within this particular community. I have no doubt that it will happen, and I think as with the emergence of whole genome sequencing and ways to impute deleterious natures of mutations within regions of the genome that are not exonic will be exciting and will be emerging soon. Excuse me. All right, so wanted to zoom in on yet another subnetwork in 2012 that involves these individuals whose word clouds are represented here. We have an individual with a great deal of expertise in DNA analysis, sequence analysis. I happen to know that Sekwon is a brilliant mathematician and statistician. We have Ingrid Holm, who has incredible expertise in clinical relevance, clinical work, and among many other things represented on this word cloud. Zach Ohane, obviously a juggernaut in the field of microarray analysis, computational biology, bioinformatics, clinical bioinformatics, personalized medicine, among many other things. Lou Kunkel, one of the arguably world's best geneticists. Putting these individuals together, we come up with something incredible for autism. A study that came out recently in PLOS One, this isn't the only one from their work, but I just wanted to highlight this one, which is the earlier one that demonstrates through rigorous analysis of RNA microarray data, gene expression patterns between healthy individuals, neurotypical individuals, and ASD to identify a diagnostic marker, a marker that is able to classify between ASD and non-ASD, a marker that has potential to be able to do this. In their work, they're able to show through a few hundred individuals that there is reasonable separation, and that reasonable separation is a bright light in an otherwise murky swamp. And this is an indication that there is a possibility that we can use RNA and gene expression patterns to be able to differentiate at an early stage an individual who might have risk for autism from an individual who does not. And so some of this helped to inspire a company that I admire greatly, Synaptics, which has come on the scene to be able to push forward the idea that RNA can be utilized for diagnostic potential. And they're doing so in a lot of very rigorous and careful ways. Right now, they're working on clinical precision and impact, and they're trialing in, I believe, 17 sites independently to identify clinical impact, to measure sensitivity and specificity, and to further develop some of these earlier ideas that came from the collaboration that I just represented. The point here is that we want to get to an area where we're going from a subnetwork of high impact, a cell paper, a neuron paper, to something that we can actually use and that delivers something back to the community. And I think this is one example in which there's high hope that it will actually go that direction, where you have incipient research that has some interest from a research perspective that gets translated into a clinical enterprise that has the potential to be globally distributed in a sustainable way, and that's through commercialization. So another collaboration here. Helen, Bill Basel, this one is particularly interdisciplinary. Bill, is a physicist essentially, and Chuck Nelson, who I know you all know well, we're one of the pioneers in a lot of areas, but in EEGs and use of EEG, and a very exciting recent paper in 2013 on that. Putting these individuals together, we wind up with another interdisciplinary subnetwork that looks like this. And it produced this incredibly highly accessed, in a very high impact BMC medicine. The BMC journals are high impact in general. This particular one is particularly high impact. A paper that demonstrates that there is a potential to utilize EEG to differentiate between ASD and non-ASD. Again, something that has real palpable possibility for translating to communities, to the community, to the bedside. So quickly just looking at that, to show you some of the results from that study. At 9 months, you have a growing difference and an increasing difference between typically developing individuals, low-risk individuals who don't have a sibling or family history of autism versus high-risk individuals who do have a sibling with autism. And there's a stark difference in the complexity of the EEG that exists here. And that difference in complexity might be enough to represent a clinically translatable biomarker after we further study it. And so I think the interdisciplinary subnetworks here, again, have worked towards our advantage in the sense that they've provided something that's at this stage research, but has great potential to translate to the clinic. 2012, another one I'm going to highlight. This is a one I know a little bit about. It's a little bit smaller of a network, but it's very interdisciplinary in that it's me working with Rhiannon, Elizabeth Harstad, and others that are not highlighted particularly in this frame. And this is, for me, a foray that's far outside my original comfort zone in machine learning and artificial intelligence, pattern matching, and systems biology. Coming into this collaboration, I knew nothing about the way in which we clinically phenotype or research phenotype individuals at risk for autism. And it's through this collaboration that I became much more well-versed, and through this collaboration that individuals like Rhiannon and Elizabeth provided me with the appropriate levels of sanity to be able to digest the outcomes that were coming from the research that we have done so far. So this is my word cloud, and I think I've already stressed, you'll see networks, genetics, numerical, reproducibility, expression. It's meant to indicate largely more numerical stuff than human first type stuff. And Rhiannon, of course, is somebody who is a professional in the diagnosis and treatment of individuals, children with autism. Together, we embarked on a research program to mine thousands of archived samples of ADOS and ADI, the two instruments that are used and considered to be generally gold standard in the field for detection and characterization and screening in some instances of individuals with autism. This is the Autism Diagnostic Observation Schedule, Autism Diagnostic Interview Revised, two instruments that have incredibly high test validity, but that are lengthy, that are time-consuming, that must be administered in a clinical setting, and therefore, as a consequence, are limited in their reach. And a question that we asked was there a possibility of reducing the complexity of these examinations without actually sacrificing their accuracy? And so through this machine learning exercise, we were able to do that in two ways. To develop one observation-based classifier that contains 8 observations, takes 5 minutes to compute. It functions on home videos, can be administered from home. Families can upload their data to a website, and we can analyze that and algorithmically produce a quantitative phenotype that indicates the individual's risk for autism. We can also take in information from a family member, a parent, a caregiver. In this case, it's 7 questions, takes 3 to 5 minutes. It's accessible, easily accessible to parents. It's happening in many, I'll tell you in a second, we're clinically trialing both systems right now. These are both right now showing to be extremely accurate, highly sensitive, highly specific. Am I running out of time? It says I have 6 minutes. I only have 30 more slides. So we have clinical trials now. So this is my way of saying this interdisciplinary collaboration, which is growing actually, working with Lenny and Bill and Bill Barbarisi and Lenny Rappaport and many others, is one that I think also has potential to be translated to clinical utility. And so as a consequence of that effort and motivated by that potential, we have clinical trials underway at the Developmental Medicine Center to be able to understand the efficacy of this rapid behavioral system, to understand where it can be used, where within the pipeline of the complexity of the autism diagnostic procedure does this particular instrument, this particular procedure have the best, highest likelihood of being beneficial. Is it upstream, downstream, can be used for clinical triage and so forth. We're figuring that out right now. We're also doing that by reaching out to families and trying to create a new paradigm for clinical validation in some ways that is crowdsourced, that simply reaches through the internet, through the fan base of Autism Speaks and other foundations, asking for research participation by the individuals who like those particular Facebook pages, those organizations. And we're getting a huge amount of traction there. And that traction is leading us closer to being able to identify a clinical solution that actually has potential, that can be done in real time. We can watch a video, score it, produce a quantitative watch a video, score it, produce a quantitative score that indicates risk, and then put that into a clinical impact report that that individuals like Lenny Rappaport can use for clinical triage to say, okay, I understand this particular kid. I can and therefore I don't have to spend the amount of time that I might normally have spent before the kid walked in the door. And so again, the hope here is to be able to put something in the hands of families that bridges the gap between those families and clinical professionals who can provide the official diagnosis. So looking forward, the Connectom has resulted in this and many other things. And it really is a portent to a future that is likely to result in real solutions, I think. One way that we might see that happening is through even further interconnections, not just with our researchers on research topics, but with what the research is producing. So if we have an EEG signature that we can utilize as a biomarker, we have loss of function mutations that we can characterize from samples that are derived from children at neonatal stages of development. We have an RNA signature that we can use. We have a behavioral system that we can use. The combination of these modalities, and potentially many others, is likely to be the right way to go. And I think it's through the Connectom and through the interdisciplinary collaborations that have emerged in the Connectom that we're going to be able to get to this point where we have a way to combine multiple modalities of diagnostic testing to produce a very definitive outcome that can be used to direct treatment and watch that treatment as it progresses. So I wanted to say, at least as a future looking forward, that's one way to go. Let's combine modalities. I think another way to go is to reach out to the network that already exists. There is a family network. There's a very large community of individuals who have faced the diagnosis of autism and who are now managing and navigating the complexity of that diagnosis, who are participating in research, who are participating in family networks, social media forums, and compartmentalized social networks to engage at various levels, including engaging in research. And so one example from my own experience is we reached out to the community to be able to validate some of the things that we have done in terms of machine learning characterization of autism. And through that, through the use of Facebook and reaching out to fans of organizations like Autism Speaks, we were able to get 11,000 participants in less than 6 months. This is a huge number in a very short time frame. This has provided us not only with proof that the community wants to help, but proof also that you can get research done by reaching out to that community. And so I recommend we do more of that, and that we have our research Connectom, we have our family Connectom, and we try to combine the two. And there's interesting emerging pieces of this already out there. My autism team is a compartmentalized social network that has, at least the last time I checked, over 44,000 people enlisted on it. And of course, Ian, which has been around for a long time, has been fostering the connection between families and research. And finally, we are doing more in my lab to try to be able to leverage this social network of families and bridge the gap between the research network and the family network in ways that are important. We recognize that our clinical solutions to provide a diagnosis aren't enough. Simply providing a piece of paper that says you're at risk or you're not at risk is simply not going to be enough. We have to be able to develop methods that are going to provide full services, that are going to enable families to really navigate through that complexity I was talking about. And so we've created a system called GapMap, which is designed to map in real time the prevalence of autism through community participation, and to be able to quantify with that map the distance between those individuals and clinical services. And then to use those quantified measures of distance between people in need and places that can provide the care to lobby for change. So this is just to show you that it's nearly live. We've been building it, we've been testing it, and I think it has a lot of potential, and I think it's a way to begin to at least quantify the gaps in clinical services and to begin to provide a mechanism for full solutions for families. All right. I have 43 seconds to acknowledge the Autism Consortium. Thank you very much. It's been a pleasure to do this. It's been an exciting and different in some ways research project. I'm not looking at nodes that are genes and proteins and so forth. I'm looking at people. It's an interesting change. I also obviously have to thank Marlena Duda, who really was the engine behind this entire effort. And so I'm just up here speaking. She's up there somewhere. She's the architect. And thank you.